Fungi. While they may look fanciful and jovial at times, they have their own dark history. Phytophthora infestans, a water mold and Protistan fungus, was the cause of the Great Potato Famine in Ireland between 1845 and 1849, a blight that resulted in around 1 million deaths. However, fungi aren't all bad. The yeast that we use to make our bread and our beer are Ascomycetes, as are the penicillium fungi that we can thank for many of our antibiotics. Today we will walk through the phylogenies of some Protistan fungi as well as the kingdom fungi to see just how complex fungi really are. Although many organisms are commonly called fungi, there are important distinctions to be made between heterotrophic protists, or Protistan fungi, and the true fungi of kingdom fungi. These two groups have many shared features, including hyphal growth, saprophytic or parasitic feeding, glycogen storage, and both sexual and asexual reproduction, not to mention coenocytic or multinucleate cells. Protistan fungi in particular lack both cell walls made of chitin and septi within their hyphae. Organisms of the kingdom fungi, however, do have chitinous cell walls, and some will have septi within their hyphae to strictly control nucleus distribution. First, let's delve into three groups of the Protistan fungi. Phylum Oomycota, the water molds, have cellulose cell walls and a complex oogamous life cycle. A diploid oogonium produces eggs through meiosis, which can then unite with sperm from an antheridium. Fertilization results in diploid zygotes that can develop into oospores, which can then disperse and germinate to form the diploid adult. This sexual cycle may continue, or the organism can undergo asexual reproduction, where the diploid zoosporangium produces biflagellated motile zoospores that disperse and germinate into new individuals. Note that, while the zoosporangium produces zoospores, it is not a true fruiting body because there are not multiple types of tissue surrounding and protecting the spores before dispersal. Phylum Myxomycota, the plasmodial slime molds, lack a cell wall on all cells except their spores, instead protecting their cells with a slime sheath. Unlike the hyphal growth of the Oomycota, Myxomycetes travel along detritus in a fan-shaped diploid plasmodium that will harden into a sclerotium when the environment dries out. During sexual reproduction, the plasmodium will divide into multiple small mounds that develop into diploid sporangia that will then produce haploid spores through meiosis. These spores will disperse and germinate into either a flagellated or amoeba stage. Plasmogamy occurs when the protoplasts of these two gametes fuse, forming a single coenocytic cell. Karyogamy, the fusion of two nuclei within a single cell, will then result in a diploid zygote, restarting the life cycle. Phylum Dictysteliomycota, the cellular slime molds, also have plasmodial rather than hyphal growth, but unlike the Myxomycota, have cellulose-rich cell walls in both their spores and macrocyst cells. In nutrient-rich environments, Dicty will exist as a free-living mixamoeba. However, if resources run out, the free-living cells will join together to form a multicellular pseudoplasmodium, or slug, that will travel to a new location, then differentiate into a tall stalk with dormant spores on top. These spores will disperse and germinate into mixamoeba. Now, let's talk about the kingdom fungi. Chytrids, like all true fungi, have chitinous cell walls. However, chytrids are unique in that both their diploid zoospores and their haploid gametes have flagella and are motile. They alternate between isomorphic generations as the haploid gametangium gives rise to anisogamous gametes, which then fuse and mature into the diploid sporophyte, which can then either reproduce asexually through the production of diploid zoospores or sexually through the production of haploid zoospores. Zygomycota, the bread molds, have a life cycle that is predominantly haploid. The greenish-black fuzzy spots that you see on old moist bread are colored by the mold sporangia, which house and release the haploid spores. Note that the sporangia is not a true fruiting body since it is not made up of more than one type of tissue. Once the spores have dispersed and grown into haploid individuals, these individuals can continue to reproduce asexually via haploid spores from their own sporangium. However, if two mating types come together, their two isogamous gametes can fuse via plasmogamy, then karyogamy, to form a diploid zygosporangium. A new sporangium will then emerge from the zygosporangium, and meiosis will result in haploid spores. Ascomycota, the sac fungi, often produce large fruiting bodies that you can see across the forest floor. The key feature of the Ascomycota life cycle that distinguishes it from other fungi is the formation of specialized cells called asci that sit within the large ascocarp fruiting body. Diploid asci will undergo meiosis to form 8 internal haploid ascospores that can be explosively released from the ascus. Each spore will develop into a haploid individual called a conidiophore that can reproduce via spores called conidia. The mycelium of the conidiophore can differentiate into either a male gametangium, the antheridium, or the female gametangium, the ascogonium. A male nucleus will travel across a trichogyne into the ascogonium, resulting in plasmogamy. The resulting dikaryotic ascogonious hyphae will persist until karyogamy results in the diploid ascus. Bread yeasts and deuteromycetes are both types of Ascomycota, but are considered fungi imperfecti because they appear to have no sexual phase in their life cycles. The last group that we will discuss are the Basidiomycota, which include Hymenomycetes, Gasteromycetes, and rusts. Like the Ascomycetes, Basidiomycetes produce large fruiting bodies that aid in spore production and dispersal. However, rather than producing ascospores within asci, Basidiomycetes produce basidiospores outside of specialized basidium cells that lie within the basidiocarp. These haploid basidiospores disperse and germinate into haploid mycelia, which will then fuse to form dikaryotic mycelia. These dikaryotic mycelia continue dividing, and each division uses clamp connections unique to this phyla to ensure the proper allocation of nuclei into daughter cells. As the dikaryotic mycelia differentiate into the basidiocarp fruiting body, karyogamy results in the formation of diploid basidium cells. These basidium can be found on the hymenia that line the gills of the basidiocarp. Meiosis will result in the 4 haploid basidiospores that we started with. Fungi are not always alone in their battle for survival. In fact, fungi form some of the richest symbiotic relationships on the planet. Lichens are a great example of fungi symbiosis with cyanobacteria or green algae. The fungus provides nutrients, moisture, and protection for the photosynthesizer and receives sugars in return. Fungi can also form symbioses with the roots of terrestrial plants, either by directly penetrating root cells in endomycorrhizal relationships or forming a Hartig net around the root cells in an ectomycorrhizal relationship to exchange nutrients for carbohydrates. Clearly, the world of fungi is vast and diverse, and the importance of these small detritivores is undeniable.